On behalf of Powell-Drescher Ovarian Cancer Research Foundation, I'd like to welcome you all to Paths to Research Advocacy. It's a webinar series where we explore the journeys of ovarian cancer survivors and scientists who dedicate their voices and efforts, raising awareness about the unique paths each one of us takes in our advocacy. My name is Susun Livingston. I'm a 25-year survivor of ovarian cancer and, more recently, a research advocate. I will be the host for today's discussion. Today's format will be recorded, live interview for about 35 minutes, followed by a Q&A where we will open the discussion to our audience for you to ask questions directly to our guest. So feel free to enter your questions into the chat when you think of them, and we will be sure to include them all in our Q&A. I think you're going to like this one. So let's dive in. Today, I have the special honor of speaking with Dr. Kate Chiappinelli, Associate Professor in the George Washington University Department of Microbiology, Immunology, and Tropical Medicine. Dr. Chiappinelli received her PhD from Washington University in St. Louis and then pursued her postdoctoral studies at Johns Hopkins University. Dr. Chiappinelli then opened up her own independent laboratory dedicated to her curiosity of epigenetic changes in cancer and how to combine epigenetics and immune therapies to fight cancer, with a special focus on ovarian cancer. Not only is she an associate professor and principal investigator of her lab, managing 12 other scientists, but she's a proud mother of 3 children. She's an avid runner. She works with high school students, introducing them to science, and performs as a member of the 18th Street Singers, a 50-voice community choir located in Washington, D.C. Thank you. Welcome, Dr. Chiappinelli.
Thank you so much. It's really an honor to be here.
So my first question for you is pretty straightforward. When do you find time to eat?
I think I drink a lot of coffee. That is mostly, yep.
It's just crazy for me to think about everything that you do. Um, Before we dive into epigenetics specifically, I thought we might start off with having you tell the audience how it was that you chose research as your career path.
Sure. So I really trace this back to having a fantastic high school AP biology teacher, so advanced biology in high school, where this teacher, Miss Fields, really emphasized the hands-on science. And so I think often in high school classes, especially in science, there's a lot of memorization of terms. And this teacher, instead of just emphasizing that, said, we're going to isolate DNA from strawberries, right? We're going to learn how to actually look at the DNA and get inside the cells, right? And that made me so interested in working with my hands and understanding the mechanisms behind how things worked. I really enjoyed that. And then through college, continued to be very interested in genetics and then became interested in epigenetics, which we'll talk about in a minute, of the control of gene expression, which was a relatively new scientific part of the scientific investigation at that time.
It was because it started pretty soon after they were sequencing the genome, right?
Exactly.
So it's still pretty young. I mean, we think of, because we were all raised during that time frame, but I didn't realize that epigenetics was just more recent at the same time.
Yeah, and the understanding of beyond just the genes in our genome, what makes them turn on or off and make changes to cells, right? So all the cells in our body have the same DNA, but then how is this turned on or off?
Okay, let's get into it. But before I start, I'm going to be a good research advocate. And every time that there's a terminology that you use that may not be clear to others, I may interrupt you with the word question.
Thank you.
Okay, so let's start making epigenetics understandable. Let me start with some basics. Most of us understand that some diseases are caused by mutations that are inherited by our parents and are present at birth, like sickle cell disease, or more importantly for this audience, BRCA1 and BRCA2. If that is genetics, then what is epigenetics?
Great, thank you. Epigenetics actually comes from the Greek epi, which means on top of or above genetics. And so basically it's saying a level of control above your DNA. And so the way that I like to explain it is that all of the cells in our body have the exact same DNA. It's the same in every different cell, but those cells need to perform different functions. So a cell that's going to be in your brain transmitting a nerve impulse needs to do something different than a heart muscle cell that needs to make your heart beat. And epigenetics is how different pieces of that DNA are turned on. For example, a piece that would make the nerve cell transmitted impulse is turned on in the brain but off in the heart. And then a piece that would make the heart muscle beat is turned on in the heart but off in the nerve impulse. You can think of it like a traffic light, a red light or a green light.
So I was listening to a bunch of, I was doing some study on epigenetics in preparation for today, and I heard something just yesterday that I thought was quite interesting. It's almost like DNA. This is easy for me as being in the world of computer science.
Hmm.
DNA is like the hardware, and epigenetics is like the software.
Yes, that's a great analogy, actually. Yeah. So what's there in every cell that is very hard to change, actually epigenetics is much easier to change. It's much more dynamic.
So, because I know you're a musician, I also heard another metaphor that I'm going to test on you and see if you want to expand on it a little bit. In music, we have lines and dots with little flags on it, the notes. DNA is like a sheet of music, but then every person who goes down to play a specific piece on the piano or sing it, epigenetics is the interpretation or this special unique expression of how that music is played. Does that kind of resonate with you? That kind of description really helped me kind of grasp
Hmm.
what was going on.
Yeah, I like that of thinking of the genetic code as the piece of music and then epigenetics being how it is played in each different cell. I really like that.
Good. Um, so how is epigenetics changing the way scientists think about ovarian cancer development, recurrence, or treatment resistance? You can do all three or just take one.
Sure. I think one of the most clear examples of the importance of epigenetics for ovarian cancer is actually the gene BRCA1. And so you mentioned this before of there can be inherited mutations in BRCA1 that predispose you to breast or ovarian cancer, and those are DNA mutations. Beyond the mutations, you can also have a red stoplight called DNA methylation that is a mark that silences BRCA1. So it will make BRCA1 not expressed. It has a very similar effect to a mutation in the DNA, but it's an epigenetic mark. And patients that have BRCA1 methylation, the red stoplight, are more sensitive to PARP inhibitors. So that's an example of how this epigenetic changes the response of the tumor to different drugs.
So, question for you. Let me go back on the word methylation just to make sure that I understand it. Methylation is where it takes a gene and inactivates it
Yep,
or
that's right.
activates it,
Turns it off. Yeah, red light.
turns it off. Okay, so that means that not, does that mean that all genes are active?
Good question. No, no. It's going to depend on the different which cell and yeah, which genes are active in is going to differ based on what cell type they're in. Kind of like we were the example at the beginning about the nerve cell versus the heart cell. In a normal, for example, breast, ovarian, fallopian tube cell that is not cancerous, BRCA1 would be turned on and active, and then in cancer, it can be silenced by this methylation.
So does that explain why someone could actually have BRCA1 genetically yet not get ovarian cancer?
Have a mutation in it and not get ovarian cancer? Do you mean
Yeah,
that? Um,
it can't oversee that. It can't
no,
super.
it's
Okay, okay,
don't think of it as interacting with the mutations, right? So it's kind of a
got it.
separate way to inactivate BRCA1, a separate way to predispose you to cancer, unfortunately. And then, but also making that tumor more sensitive to a specific therapy.
Okay. So I think a lot of us have been kind of hearing about epigenetics, but we hear about it in terms of environmental situations like
Mhm,
food or sleep or exercise or environment, how
mhm.
these things can impact our genes. But I also wonder if some of this information is accurate or oversimplified. What claims about epigenetics in popular media today should people approach with caution?
Mhm. Thank you. I think that's a really important question. Um, Eh, my answer to that would be anything that's not backed up by peer-reviewed scientific research. So especially claims of epigenetic age based on how much DNA methylation is in your cells, right? So just be careful with those. I think that there is probably something there, but often it is overhyped or over interpreted. I think one of the most important things, well, something that has made its way into the public health sphere in terms of DNA methylation are studies that were first done in mice that showed that when the pregnant mom mice didn't get enough of a methyl donor, basically this was like something that provides the substrate for the methylation, their baby mice were much less healthy. And so this actually, a methyl donor is folate or folic acid. And so this is included in prenatal vitamins for pregnant women. Now, make sure you have enough of that methyl donor, enough of the DNA methylation. So these have been very well established, and now they're in recommendations, and like clinical trials were run on this, and this prevents spina bifida, et cetera, right? So having enough methylation while the baby is developing. So these are the things that have been validated and that are being acted upon.
Okay, okay. So for survivors that are listening today and non-survivors, by that I mean caretakers as well, because we consider those advocates as well, why is it important that they should understand about epigenetics, even though it's a little grokky, a little hard to understand at the scientific detail? What is it that, how should it affect our daily lives?
Sure. I think that epigenetic therapies are very exciting because they can change the way DNA is packaged rather than the actual sequence of the DNA, right? So it is potentially easier to manipulate the way the DNA is packaged and expressed. Um, a lot of these are in clinical trials right now. And so I think it is important to be able to understand it broadly the way that these drugs are working, and that can give you insights also into the effects on the tumor, but also potential toxicities or effects on other cells. And so I think understanding the basic concepts of that any drug that is called an epigenetic drug will change how genes are expressed. It will usually not be targeted. So there are some therapies that are targeted to a specific gene. When you give someone an epigenetic drug in general, you are affecting the whole genome, potentially the whole genome, maybe some locations more than others, but that's also really important to understand. And then finally, we and others have shown that epigenetic drugs can initiate anti-tumor immune responses through a couple different ways. And now they're being combined with different immune therapies in clinical trials. So not just alone, but with different immune therapies in clinical trials for ovarian cancer and other solid tumors.
So let me unpack that concept of epigenetic drugs and immunotherapy. So let me go
Mhm.
back to epigenetic drugs for a second. What are epigenetic drugs?
Hmm. These would be drugs that either in with the that usually inhibit enzymes that add like a methyl group, right? So the enzymes that add the epigenetic marks and change to a red light or a green light.
Okay, okay. And then that in combination with
Mhm,
immunotherapy.
mhm, mhm,
Okay, okay. All right. So if I understand you correctly, oh, let me ask you about another question. This is interesting to me, this reversibility. Let me see if I can do my best to explain it.
mhm,
You can help me even do a better job, I'm sure. Um, Epigenetic changes for me seem to offer some sort of hope for some cancers, not all cancers, right? Because epigenetic changes can be reversed, while DNA sequence mutations like my BRCA1
mhm,
cannot be
mhm.
reversed in any easy way. So are we early on with these epigenetic drugs to be able to reverse some of these red lights and green lights, as you call them?
Great question. I think that right now some of the drugs we have don't work as well as we want them to. So I think there's a couple different issues. So in the case of DNMT, DNA methylation inhibitors, that will erase DNA methylation, um, a lot of the clinical trials right now are using different formulations of the drugs to make them more stable. So it's not even mechanism, it's how well is the drug working, right? So there's a promising trial in Italy combining DNMT inhibitors and immune therapy for lung cancer, a promising trial in melanoma, a promising trial in lymphoma. And I'm hoping we can get there for ovarian cancer as well with a combination of this and different immune therapies.
Okay. All right. Woo. Wow. Thanks for helping us try and demystify this.
These are great
This
questions.
it is well, and it's a tough topic. Epigenetics. I mean, I think most of us know about genetics because we're getting really comfortable with the concept of, oh, I mean, somebody's diagnosed, and I immediately ask, do you have BRCA1? Do you have BRCA2? You
Great.
know, it's like this is
Mhm.
now part of my language, but the concept that there's more above and around the genes that can control, that can make a difference is like a whole new kind of mind-blowing experience for some of us, at least.
Yeah. Yeah, and let me clarify one thing on the BRCA1, just so I want to make sure I'm not misunderstood here. So as you said, many people inherit one mutated copy or deleted copy of BRCA1, right? And then it's the other copy gets a hit or a mutation, and that's when cancer develops, right?
Uh-huh.
The BRCA1 methylation is usually happening in the tumor. That is in general, it's
Oh,
going to
okay,
happen,
okay,
right? So it could be a second hit actually, where you have a BRCA one of the copies is mutated, and the other one becomes methylated.
okay. All right.
Mhm.
That's the reason why I'm not a scientist.
It's hard. Yeah.
This has been incredibly helpful. I'd like to shift gears a bit and talk about one of my favorite topics, advocacy. At the Powell-Drescher Ovarian Cancer Research Foundation, Kate, as you know, we believe that connecting scientists and survivors can improve both research and patient outcomes. Advocacy is a very broad term, means a lot of different things to different people. So let's begin with a simple question for you. What does advocacy mean to you in your role as a scientist?
Thank you. Um, yes, I think that's a really good question. I think one thing I try to do is interact with more ovarian cancer patients, survivors, and advocates to understand more from their perspective. I think it is important for people that fund the research, so that would be private foundations, NIH, DoD, et cetera, to understand the impact of it. So I am always, I mean, I'm in DC, right? So I always volunteer if they want some testimony from a scientist to show here are basic mechanisms that have been translated to clinical trials, et cetera. So that's what I try to do on my side of things. In terms of the clinical side, I have colleagues who are OBGYNs who will, if they have someone who is delivering a baby by C-section and they know it's going to be their last kid, they know to say, do you want to remove your fallopian tube? So things like that are in terms of prevention as well. So I think there's many different ways we can be advocates for better understanding of the disease and then for getting more research funding for it as well.
The other thing that I found interesting, you and I happened to have the luxury of having breakfast together down in San Diego when we were attending the American Association of Cancer Research um, annual meeting. And one of the things that kind of struck me was that you actually, as a scientist and as a friend, are more than willing to go with your female friends on the appointments to talk
Yes.
about some of these tough topics. I was so impressed with that.
Oh, thank you. Yeah, I think you shouldn't need a PhD scientist with you at your appointment, right? Like I wish we were living in a world where things were better explained and then that patients could be their own advocates. But sometimes it's really helpful to have someone who knows more about it in your corner. Yeah.
So I know even recently I was even asked, what is there's so many different ways that a survivor or a caretaker can actually enter into the world of advocacy. Sometimes I feel like I define it too narrowly because I'm so passionate about research. And so, but there's so many different ways. What forms of advocacy do you see that go unrecognized that are still of high value?
Good question. I have really appreciated when an ovarian cancer patient, survivor, caretaker, advocate comes and speaks to my group of scientists. We actually, I think I see Anna Haddon on the call. She came to visit my lab actually, maybe a year and a half ago, I think, when she was in DC for a meeting. And it was really valuable because I think that sometimes there is not enough interaction between the scientists who are working on the disease and the people who are actually dealing with it, right? And so to hear the perspective is really, really valuable. And I think we, as scientists, we benefit so much from your perspective and questions for us and just interaction with us. I think people really see a concrete output for all of the hard work that they're doing.
Or as my scientific mentor, Dr. Kristen Anderson, always reminds me, they're as afraid of you, Susan, as you are afraid of them.
Oh, absolutely. Yeah. Don't be afraid of us.
So many survivors deeply admire scientists and researchers, but feel intimidated. The language, the education, the terminology. Why do you think that gap exists? I mean, those things I've mentioned are part of the gap exists. But what can we do to minimize it? I mean, what can we do to kind of get us all on the same level, and knowing that we all wear pants and put our leg in them at one time at a time, one leg at a
Mhm.
time?
Yeah, absolutely. I think there's no reason to be intimidated. I think that unfortunately there's a lot of jargon, right? Like we talked about. So even explaining our research, I could use six different words that no one would have heard before, right? To describe it in one sentence, right? So I think there's number one jargon, but there's a way around most jargon. Like every scientist should be able to explain their work in plain language. And Also, what we try to do, another thing we try to do with my lab is do outreach in our community. And so specifically, this is high school students. So we do outreach both in terms of explaining really DNA and epigenetics, and then how does cancer happen, but then also working with them to do experiments and say, you can be a scientist. This is a career you can do. So we try to start on the early side in terms of both visibility. I have my trainees, the PhD students or fellows, explain to the high school students how they got there. And it's not often a linear path, right? It doesn't have to be, oh, I got straight A's in genetics in college. And so then I went right to grad school. It can be, I didn't know what to do after college. So I worked in a lab for 2 years and I decided that I really liked this. And then I went into this. So we try to approach it from that side of making it more accessible. And then for me, having the trainees then have to explain in to the high school students what they do, the science of it keeps it relatable.
And it's interesting because when I go to scientific conferences, I'm now much more comfortable. But more
Hmm.
importantly, I've actually developed a curiosity to ask the question of the researcher, how did you get here?
Great.
And
Yeah.
they're all so different stories. I mean, we always assume, like you said, it's I got straight A's, etc. I love it when I talk with the researcher and they say, I have had cancer, or my
Mm-hmm.
mother died of cancer,
Hmm.
or I saw my sister die of cancer.
Hmm.
And so these are I think part of the thing too is us just getting the courage to just like we would at a cocktail party. Who are you? Where do
Hmm.
you come from?
Absolutely. Yeah. And I mean, to that point, I appreciate that you asked. That's really great. I'm realizing that scientific conferences are probably some of the most unintelligible talks because
Oh,
people use a ton of jargon and then they speed through their talk. So I think it's things like that we can then, it would be great to have some push to make those a little bit more accessible. For example, when you attend the ASCR meeting, to be able to get the most out of the scientific sessions.
yeah, it was great. I love it. I can't wait to go back. Can you share a moment when an advocate changed the direction of your thinking, your research, or your understanding of what matters most?
Thank you. I was fortunate. I was funded as a member of the Ovarian Cancer Academy from the DoD, and basically this is an early career grant that's given to new investigators working on ovarian cancer. But more importantly, it is really kind of a career development community where you have the early career investigator and the more senior mentors. And we have a conference about once a year where we present data. We do professional development things. And this finished for me 2 years ago, but it was incredibly valuable to me. And I have a lot of collaborators from it. I've learned so much. And at the one that was 2 years ago, I was moderating a panel with ovarian cancer patients and survivors, and we were discussing what are the barriers to getting on clinical trials, right? Thinking about when if we are fortunate enough to have something that we're really excited about in the lab pan out and get to the phase 1 clinical trial, why do you choose to get on this trial or not? What are the risks and benefits? And a lot of the things they brought up, such as off-target effects, how often you had to come in for the treatment, things like this were things that I had not thought of and I really should have thought of. And it really helped me in terms of if we're really trying to help people coming up with new therapies that are just much more realistic and don't require such a heavy lift from the patients.
I actually did participate in a clinical trial, and I feel so excited about clinical trials. And it's so weird when I talk with other advocates because they're so scared when they're being offered a clinical trial.
Yeah.
And I always go like, oh my gosh, jump on the bandwagon and get excited because you're getting exposed to some of the latest and greatest things that are just so exciting. There's so many things that are in the pipeline. And that as patients, we're terrified of the word clinical trial because we think it means, oh, you're really in bad shape.
No, yeah, yeah, it's a really good point. It's like the only way that we move forward, and I think we really need new therapies for this disease, right? And so this is the way to do it.
Absolutely. What kinds of questions do you wish survivors would ask researchers more often and why?
Oh, I would love to get more questions about the just how the things we're studying work. And then that we can, talk about that and learn together. I think that would be great. I think you always learn something, especially when new voices ask you a question, right? And it makes you think about something in a different way.
On that note, what are some of the most exciting things you're working on right now today in your lab that just kind of make you jump out of bed to go there?
Thank you.
And you can translate into regular layman's language.
Yes, yes. Okay, so we talked about methylation and epigenetics, and and in general, there's a lot of excitement about immune therapies overall, of therapies that can activate our own immune system to eradicate cancer cells, just like they eradicate cells infected by a virus or a bacterium. Our lab connects those two in kind of an unexpected way, where we discovered that one of these DNA methyltransferase inhibitors, one of the drugs that removes the red light on the DNA to turn on genes, also turned on expression of pieces of our genome that were not thought to code for genes. So only about 2% of our genome makes genes. The rest of it was termed junk DNA by the Human Genome Project. We're finding out that it does a lot more. These pieces make RNA, so DNA makes RNA, makes protein, and the RNA looks like it came from a virus. It looks like viral RNA in our cells. It's not actually making a virus, but it kind of fools the cell. And the cell thinks it has an infection and brings in immune cells to kill the infection, which is the cancer cell. And we called this viral mimicry.
So it's kind of like teaching the body to be smarter.
Exactly. And teaching, in this case, making the cancer cells bring in immune cells to kill them by activating this signal, but coming from this epigenetic change in the DNA.
Woo.
Yeah, it's a lot. And right now, that's kind of the basic premise. Right now, we're learning much more about how that works and how we can make that work in different tumor types in combination with other therapies, et cetera.
So you have 12 scientists in your lab?
Yes. Not
Wow.
all the time. Some are undergrads. Some are like 12, 10-hour a week undergraduates, but yes.
And what brings them into the lab? I mean, what's their story? I mean, how did they get into this? Any great stories that you have about your own lab rats?
Let's see. I wish I did. It's a group of either postdoctoral fellows, so after their PhD, or PhD students, or undergraduates. And And I have a lab manager as well and a research technician. And so everyone is, I mean, they're just all they want to be scientists. They are scientists with an interest in either epigenetics or immunology. I do think that our group benefits from this interdisciplinary interaction in that we have some people who are much more immunologists and some people who are much more genetics, epigenetics people, and that we then keep each other on kind of honest, right? Of making sure that we're doing both well. Yeah, and the model is that, we'll have undergrads working about 10 hours a week as part of their biology major, and I usually pair them up with a PhD student their sophomore year, so we get them for 3 years. And then they do a real substantial part of work and they are part of the papers and everything. So they do quite well.
Do any of your do any of them actually have had cancer or have been impacted by
Hmm.
cancer?
Let me think. I've had a fellow or two who had previously had cancer. The PhD students, many of them have family members or close family friends who have had cancer, and so this is often a motivating factor for them to be in cancer research.
Great, great. So, as you can see in the chat, I'm going to start to shift gears. We're going to do a closing question in this one area, and then I'd like you to either open up your mics and ask questions in just a few minutes. But before we open it up for questions for our audience, I have one last closing question for you. And that is because there are so many changes of late, specifically around federal funding, don't get me started, federal funding for research being so dramatically reduced, what gives you hope, whether it be science related, advocacy related, or something else?
Thank you. It's a great question. Um, the I think it has been a very challenging environment in the past year and a half. There have been very important wins as well. So I think what I have seen is that last year, I'm trying to think about a year ago or so, there was a lot of successful advocacy. There was a potential 40% reduction to the NIH budget, and that did not happen, right? There was like a 1% to 2% increase instead. There are a lot of very bad things happening right now in terms of rules from NIH. Changing the way that grants are reviewed and funded. So changing how the review is normally, grants are how I get money to do the experiments in my lab, and they're competitively reviewed by my peers, other people who have expertise in genetics, epigenetics, immunology, and this would change that to review by a second layer of review by political appointees. And then also rules restricting how many grants any one individual can have. And so there is public comment available on these rules, and a lot of people are putting in public comments saying that this would not be the best way to fund the best research. And so I think advocacy is really important, both from survivors, from patients, and scientists across the board.
Okay, you heard that cry for advocates to be able to speak up. I don't, I've got a few questions, but I don't see any. Please, if you have a question for Dr. Chiappinelli, also known as, I refer to her as Dr. Kate,
Just Kate is fine. Yeah.
feel free to unmute yourself and ask the question. Raise your hand if you would so that I can see it. Um, I would like to kind of keep on this train of thought. What is your perspective on AI and its role in the world of gynecological cancer research?
Mhm. Um, I think in general, um, the that there's a lot of potential in terms of deploying AI to perhaps identify novel targets, to be able to analyze large data sets, right? So things that we do in my lab, we are often analyzing large data sets. So what do I mean by that? That's like we sequence the whole genome of a person with ovarian cancer, and then we sequence what's being transcribed, that layer after, the RNA coming from that genome. A lot of that you can use, and my trainees have used AI to help make that faster, right? So that's a kind of on a small level, is that you can do analyses that would take you longer. You can do it in a shorter amount of time. Overall, there's exciting you could think of that then on a higher level of applying that across multiple different sites to learn more about the disease. So I think there's a lot of potential there, and we should just be thoughtful about, privacy of information and things like that, that with any new technology, right? And so, approach it cautiously and understand how it can actually help you move the research forward.
One of the things that I learned when I was down in San Diego was I was so blown away with how much AI is coming into play everywhere in research.
Mhm.
And one of the concepts that I fell in love with was this whole idea of molecular sequencing of a tumor.
Mhm. Mhm.
And that AI data being able to actually be used in influencing on the treatment of that
Yeah.
patient.
Or even liquid biopsies where you are not even doing a biopsy, you're just taking the blood of the patient, and then in the blood there are pieces of DNA from the tumor, right? So that's even better. It's less invasive, and then that's already been shown to really, uh, predict response to therapy and progression, right? So these are all this is very exciting.
And what is that called? Is that ctDNA?
Yeah, exactly. Circulating tumor
ctDNA.
DNA. Good.
Right, right. We do have a question that's come in, and I'd like to ask it on behalf of them, whoever privately. Is the research community getting any closer to a diagnostic for ovarian cancer?
Great question. So it's so needed. This is not something that I personally work on, and I'm just going to say what I know from conferences. That there are, I actually related. We talked about that only 2% of our genome makes proteins. One part of the genome that's not that is called line 1, and this is something that is expressed at higher levels in ovarian cancer than pretty much anything else. And this is really spearheaded by Ronnie Drapkin, who maybe you're familiar with, and Kathy Burns, and many other colleagues. They are moving into clinical trials to test both its level of line 1 protein in the blood as well as other markers. So it's not just that, but in combination with other ctDNA in the blood to be able to diagnose this earlier. And then I think also testing as to be used as a predictor of response to therapy or indicator of response to therapy. So that is the most recent information I have. I think it's really needed, and the using this protein that's overexpressed in ovarian cancer, I think is very promising.
Good, good. Another question's come in from Deborah Binder. How can we create more of a sense of urgency for a better diagnostic test for OVACA? I always say that if men had ovaries, we would already have a test. I think that's a good point.
Fantastic. Thank you, Deborah. Nice to see you. Yes, I think it's a really good question. When I explain to people, I think, again, more advocacy, because when I explain to people, say, oh, okay, well, we have screening tests we have screens for prostate cancer, we have colon cancer screening, we have breast cancer screening. Not that any of these are perfect, but they really reduce the burden of disease, and explaining that there's really nothing, right, for this disease. So I think advocacy, and I think recently, it was several foundations just funded new projects, all focused on ovarian cancer diagnostics. So I think that the work is being done. I think right now it has to be, going into trials and being validated.
Great, thank you. From someone who's actually been to your lab, and I'm envious of it, Anna, how can our community collectively ignite the light behind the critical need for rare tumors in ovarian cancer?
That is a great question. I think that there is less so how can we, promote understanding of the different biology of these rare tumors? I think that there can potentially be a lot more that we don't understand there. And so I think education is the first thing to understand the biology of these, and then that could open some doors in terms of treatment. But so in terms of how can we then promote knowledge of this and for example, not only hear about high-grade serous ovarian cancer all the time. I try to be thoughtful when I'm giving a scientific talk of showing there are many different types of ovarian cancer. Here is what I'm focusing on right now. But I think it's a really important unmet need.
I agree that I think that there's a lack of knowledge really, unless you're in it, of how many different subtypes of ovarian
Mm,
cancer is and how small those subtypes are, thus therefore immediately increases the complexity of being able to find a solution for it.
hmm.
So it's a tough challenge. Back to another question. Will some of your research potentially help with effective screening for ovarian cancer, or are you more focused on treatment and hopefully a cure?
Oh, great question. Definitely more focused on treatment and curative immune therapy is the goal. But I think some of the research I discussed previously, which others are doing, again, focused on this kind of the dark genome or the parts of the genome that we don't know as much about that are actually at higher expression in these cancers are very promising.
Question, dark genome.
Yeah. It's kind of a layman's term, basically saying that we understand 2% of the genome. You can think of that as the light, and then the dark genome is the parts that we don't understand what they do, and that we're just beginning to learn more about.
Okay. Deborah Binder's following up with her previous question.
Mm,
Her previous question is, how can we create for better diagnostic tests? Her follow-up question is, why is it so hard to find something that works for
that's a great
ovarian?
question. Yeah. So I think, just thinking about the history of this, right? The trial in the UK, where they combined CA-125 and ultrasound, right, did not diagnose, more ovarian cancer, save more lives. Um, I think this, in terms of why is it hard to find a diagnostic that works, I think we face the problem with cancer that cancer cells are not that different from our normal cells. And so how easy is it going to be to detect something in the blood from an early stage tumor that's different enough, right? Maybe you'll detect it from a stage 3 or a stage 4 tumor because it's shedding more DNA. But can we get down to that stage 1? Is there something that's different enough? And ovarian cancer, unlike a lot of other tumors, it doesn't have a lot of specific genes that are mutated, right? So you have P53, a tumor suppressor that is mutated in most ovarian cancers. You have BRCA1, BRCA2, and there's not a lot of other mutations that are really distinctive of this, right? And so I think it can be really hard to identify what is getting into the blood from these early stage tumors that can then be used as a diagnostic.
So, kind of pivoting it back onto the concept of advocacy, for someone who's listening today who wants to get involved with research advocacy, what do you think are the first good steps?
Hmm. Um, I mean, I would say come talk to researchers, and I'm happy to connect you, with my lab. I think it'd be great if you can. I don't know. I think it works well to find someone that is in your town or close to your town, so somewhere. But you can also connect. Like if the only way to have a survivor speak to us would be virtually, we would totally have you do a lab meeting for us virtually, right? Where you would then the person would tell their story, and then we could ask them questions and talk about the different research we're doing. One thing we started here was having interacting with the hospital and the ovarian cancer patients, having people from my lab do like a 10-minute talk on what they were working on and making it open to, we basically just emailed all the gynecologic oncology patients and providers. So anyone who wanted to could come. So we would have patients, we would have their caregivers, we would have the physicians, the nurse practitioners, et cetera. And we got really different questions than we do from other scientists. I think we really benefit from that. That is good for everyone.
That's great. I have another question for you, come from the audience. You mentioned that you have trainees in your lab at many levels. What does training scientists look different? How does training scientists look different at different stages? And how do you determine who in your lab works on which thing?
Oh, great question. I really enjoy this part of the job. And really, I would say that the more senior someone is, the more skills they already have, the less you have to do as a PI, right? So a postdoctoral fellow already has a PhD, has already done a lot of training, usually will come in saying, I'm really excited to join your lab. I want to work on this, this, and this, right? And say, like, here are the questions I want to ask. I do very little. If it is a successful postdoc, like I will provide the money and give feedback. This person will come present for me once a week. We'll go over direction. I give feedback if something's not working or say, okay, this piece of data makes sense, but I think I need to see a little bit more to convince me that this is what's actually happening, right? Then PhD students, it's really going to, when they start in the program, they, are highly qualified, get into a PhD program, but early on need a lot more guidance. And then later on, as they become much more independent, are really running their own project. In terms of who works on what project, I'm pretty open. We have a couple of lines of investigation that are funded by grants that we obviously need to get work done on. But if someone comes to me and says, I've always wanted to work on this thing that is close enough to what we work on, say, go ahead, try it. Let's see what this happens. And that's happened with several of my students. They just propose their own projects. So it's really great. So that's what we do in general, and it works out very well. With the undergrads, they usually work with the PhD students, so that's kind of their daily supervisor. I still do meet with them once a month and then make sure that everything is on track.
Cool. Got another question. You mentioned that only about 2% of our DNA codes for genes and the rest are used to be considered junk. What are some of the most exciting things you're learning about the junk or the dark genome?
Great. Yeah. Awesome question. I love the dark genome. One is this line one that is 20% of our genome, 20%, and usually has this red light silence on it in almost all normal cells. But in ovarian cancer, about 75% of ovarian cancers, green light, it's turned on. You can see it making a protein, you can see RNA and everything. So for diagnostics, that's very exciting. The second is this mimicry that we discovered, where when you treat with these epigenetic drugs to remove the stoplights, you get this activation of immune responses. Third, there was a really exciting paper recently, it was in kidney cancer, that some of these can make little pieces of proteins that can be presented. So when we think about our cells normally, we have molecules on the surface that present pieces of proteins for our immune cells to go by and scan and say, oh, that's a normal cell. I'm going to leave it alone, right? In this case, this is showing I'm not normal at all. I'm presenting stuff from the dark genome, and the immune cells recognize it and kill it. And so that's like a different piece of this response, and that they're actually making these little proteins that can be presented, and they actually made a T cell receptor, basically a T cell that can recognize this in kidney cancer and kill the kidney cancer cells.
Well, that's kind of cool because then, if I understand you correctly, it sort of feels like the dark genome's getting smaller and smaller.
We're learning more and more
We're
about it.
learning
Yes,
more and more
exactly,
about
exactly.
it.
We're illuminating it. We're illuminating it.
We're
Yes, exactly.
illuminating it, right?
That
It's
was
like the
that
dark
was our
side of the
Cancer Grand
moon.
Challenges, our unfunded Cancer Grand Challenges team, but we keep going. Yeah.
Well, that's great. That's great. Well, listen, um, Kate, I cannot thank you enough. Personally, I was fortunate to have met you several years ago. I was just one of those advocates you called and said, would you be my advocate on this? And I went, yeah, yeah. And I heard about you through Powell-Drescher. So I'm just incredibly thankful for your being here today, and especially for your insights in translating this kind of murky world of epigenetics. You have brought so much enthusiasm, knowledge, and hope to all of us here today. It's genuinely been a pleasure. I hope you come back.
Thank you so much. No, it's such an honor to be here. I really appreciate all of the questions. And if any of you all on the call have follow-up questions, because these are pretty complex topics, like please, if you can share my email with them, maybe if people have specific questions and want to follow up.
That sounds great. Thank you so much. I'd like to ask our audience to take some time to complete the survey that we have in the chat, as your feedback's really important to helping us make these webinars valuable to you. Thank you all for joining us today on Paths Advocacy. Advocacy doesn't end with conversation, it begins with action. And if today's discussion has inspired you, I encourage you to take the next step. whether it's signing up for a research advocacy program, sharing your story, or connecting with an organization like Powell-Drescher that supports survivors, just remember this: your voice really matters. To learn more and get involved in research advocacy, visit our website, get involved. You can contact me as well. Let's continue to make an impact together, and let's put this disease more in the back of the seat, not in the front of the seat of our lives. So thank you so much for your time today. That's a wrap.
Thank you so much.